transported into the cell, and almost immediately upon its entry into the cell, the glucose molecule is modified. At the beginning of the quiz tonight, first question, there was a question there about the molecular weight of C6H12O6. Glucose. Glucose is a 6-carbon sugar, and each of those carbons is numbered 1 through 6. So here I'm just going to put G6P. So let me explain. The G stands for glucose. The 6, let me back up. The P stands for phosphate. And the 6 tells you which carbon was phosphorylated. So when we say phosphorylated, we're talking about adding a phosphate onto a molecule. The other thing that I want to mention to you now is that, and you're going to see this here, but you'll see this more and more as we move through the course, is that principle number 1, anybody tell me, what was principle number 1 that I identified for you in terms of the first one? Structure determines function, that's exactly right. And it turns out that your cells, I shouldn't say instantaneously, but I'm going to say it anyways. And you'll understand that what I mean is very, very, very quickly. Your cells can phosphorylate or dephosphorylate proteins, and in doing that, that changes the cellular physiology on a moment-by-moment basis. I'm not going to do my raging maniac experiment where I come along and I jump in front of somebody here, try to scare the little jeebies out of them and scare some of you, and all of a sudden you find out, oh man, your heart's starting to pump, and all of a sudden the heart rate's increasing, and that happens that quick. How'd that happen? Because we dumped a whole bunch of norepinephrine onto the heart, which modifies proteins within the cardiac cells and gets those cells to contract with more force. Happens that quick. So one of the key strategies that our cells use to modify their function on a moment-by-moment basis is phosphorylating, sticking the phosphate onto something, or dephosphorylating, removing the phosphate. And that's going to be a critical story here. Now, as I mentioned, when glucose comes in, it's almost immediately phosphorylated. What am I saying? We're adding a phosphate onto the number 6 carbon. Everybody with me on that? Now, that reaction happens pretty quick. And all of you would tell me that typically chemical reactions don't occur very quick unless there's something there to facilitate. What is that something? Enzyme, a catalyst, something that speeds up the chemical reactions. And enzymes speed up chemical reactions by orders of millions of fold. 10 million, 100 million fold. And there's an enzyme. Now, here we've got the intracellular compartment of the cell. And the fluid portion, I'm hoping all of you would tell me we refer to as the cytoplasm. So I'm just going to write the word over here, and you're going to understand that this reaction here is occurring within the cytoplasm. And there's an enzyme that catalyzes this reaction, and the name of this enzyme is hexokinase. Quick time out. And this is something that many of you already know, probably not all of you know. And that is, anytime you see a suffix of ASE, you know that this, number 1, is a protein. There are many different categories of proteins, and that ASE not only tells us it's protein, but it tells us that this protein belongs to a subgroup of proteins known as enzymes, right? It's a catalyst. It's speeding up the chemical reaction. Now, listen carefully. this term, kinases, that's a special term that is used to identify enzymes that phosphorylate target molecules. In this instance, or in this case, the target molecule is glucose. So hexokinase's target molecule is glucose. And what do kinases do? I just told you. They phosphorylate their target molecules. And by doing this, now the glucose is trapped within the cell itself. So this is a key mechanism that prevents glucose from being able to move back out of the cell. Now here's an ironic aspect to this story. Is this story that I'm describing to you is all about making ATP, and yet this first reaction uses ATP. So this reaction here burns up a molecule of ATP, so I'm just going to write ATP is broken down into ADP plus phosphate. Well, glucose 6-phosphate is modified into fructose 6-phosphate. And then fructose 6-phosphate is modified into fructose 1,6-phosphate. So we've got glucose, we phosphorylated the number 6 carbon, we now change glucose into fructose. That number 6 carbon is still phosphorylated, so that gives us fructose 6-phosphate. But now we've got this other chemical reaction which converts fructose 6-phosphate into fructose 1,6-phosphate. And what that means is we've added a second phosphate to fructose. And that second phosphate now is bound to the number 1 carbon. Yeah?
So from F6P to 1,6, you're adding another phosphate.
That's correct. And you would tell me that we're able to add another phosphate via what?
Hexokinase.
Via some type of a kinase, right? Not hexokinase, but I like your answer. Very good. In this chemical reaction of converting fructose 6-phosphate into fructose 1,6-phosphate is catalyzed by an enzyme known as phosphofructokinase. We're going to refer to this as PFK. And I'll come back to what I said earlier. This story is all about making ATP, and yet at this point, now we've actually had to expend 2 molecules of ATP. So here, the conversion of fructose 6-phosphate into fructose 1,6-phosphate costs us another molecule of ATP. So at this point, we've consumed 2 molecules of ATP. By the way, there are several people in here who've had biochemistry. They know these stories very well, and they'll tell you that what I'm showing you is a gross simplification of this story. So you might sit there and feel overwhelmed by this, but just appreciate I'm giving you a skeletal outline of everything that's going on. Well, eventually fructose 1,6-phosphate is converted into pyruvate. This conversion of a fructose 1,6-phosphate into pyruvate does a couple cool things for us. Here we have, for the first time, the makings of ATP. So we have 4 molecules of ADP plus 4-phosphate reacts to form 4 molecules of ATP. So now at this point, we've actually got a net production of 2 molecules of ATP. We produce 4, but the cost is 2, so the net is 2. And I'll come back and emphasize a critical point here in a moment. Well, let's see here. Elizabeth, this story is all about what? The story of cellular respiration is all about what? Okay, but not quite the answer. What's that? No. Take a look at those 8 fundamental principles. There's a word in there that keeps popping up.
Using oxygen to make ATP.
No. Electrons. The story is all about electrons because electrons possess the energy. Remember, I talked to you last week about electron ladders. So where are the electrons? That should be your question. You should be going, hey, Dr. Cayozzo, you said the story is all about electrons. I don't see those. And you said that we need to have these electron carriers. I don't see those. The conversion of fructose 1,6-phosphate into pyruvate leads to the reduction of 2 molecules of NAD. So we've got 2 NAD, we're going to say this is the oxidized form, plus 4 protons, plus 4 electrons, reacts to form 2 molecules of NADH. This is a reduced form of NAD plus 2 protons. Just to be clear, of these 4 protons, 2 of them are here, and here are the other 2. And these other 2 are out here in solution, just floating around, and as is the oxidized reduced forms of NAD, just through random molecular motion, moving around, bumping into different things. This breakdown of glucose into pyruvate is known as glycolysis. Glyco, sugar, lysis, breakdown. I'm going to come over here because I've kind of put myself in a little jam here. That pyruvate, I'm going to extend that out over here. So hopefully everybody understands what I'm doing. Here's our pyruvate, and we've got this arrow coming off over here. And in the absence of oxygen, so up above this arrow, I'm going to put W/O, O2. So you're going to interpret that to mean without oxygen. Pyruvate, in the absence of oxygen, is going to be converted to lactate. Now, some of you may have heard the term lactic acid or pyruvic acid. Lactate and pyruvate, those are just the ionized forms of. So pyruvate is the ionized form of pyruvic acid. Lactate is the ionized form of lactic acid. You can use those interchangeably. I'm not going to say anything about that. Okay, now, so just to be very clear, when glucose is converted to pyruvate, and in the absence of oxygen, it's going to be converted to lactate. And this process of converting glucose into pyruvate and then into lactate, we're going to refer to as anaerobic glycolysis. The word aerobic means with oxygen. And hopefully all of you know that the prefix "an" means without. So anaerobic simply means without oxygen. So down beneath the arrow here, I'm going to write anaerobic glycolysis. And just let me repeat myself here, so I'm trying to be as clear as I possibly can. Anaerobic glycolysis is a conversion of glucose into pyruvate, and in the absence of oxygen, that pyruvate will be converted into lactate. That whole process is anaerobic glycolysis. Everybody with me on that? Okay, so now, with anaerobic glycolysis, what do we get? Well, in anaerobic glycolysis, we burned up 2 molecules of ATP and we produced 4. We have a net of 2 molecules of ATP. Now, I'm just going to give you a heads up. That's not very much ATP from a molecule of glucose. As you're going to see, when we have oxygen present, we're going to get a yield of ATP that's going to be up around 36 molecules of ATP. Let me just stop here for a moment, ask if there are any questions. Please don't be bashful speaking up. Yeah, go for it.
So, on the right-hand side, we see NAD also positive go to NADH. It's because it's the hydrogen atom became a negative carrier. So
Don't even worry about it.
go with that. Okay.
All we're worried about is that we've got these oxygen carriers and they're going through states of oxidation, or being oxidized and being reduced. There are carriers. They're like somebody delivering the mail, right? They come in, they offload the mail, they go get some more mail. In this case, our mailman is capturing electrons, delivering them, and then going to go find some more electrons. That's the critical part. Go ahead, speak up.
So fructose kinase, that is used to turn glucose into fructose,
Yes, speak up.
does fructose kinase, that's used to turn glucose into fructose, or completely off? Or to break down?
You're asking, does what convert what is what?
Fructose kinase, what is it exactly?
The phosphofructokinase.
Phosphorus,
Yeah.
yeah.
Yeah, the phosphofructokinase, PFK, is converting fructose 6-phosphate into fructose 1,6-phosphate. Here, through the activity of hexokinase, we added a phosphate onto the number 6 carbon. Here's that phosphate that was added to the number 6 carbon, but now all of a sudden, as kind of implied through this nomenclature here, now you see a 1, 6. There's the first phosphate that was added, and now through the action of PFK, we've added another phosphate to the number 1 carbon.
So in the first conversions, we're using hexokinase and PFK, and then from the fructose on 6, we're converting that to pyruvate via the oxidation reaction.
So,
Like we're using catalysts in the first two conversions, and then the last, and then from fructose, we're adding the phosphate, converting that to pyruvate via the oxidation reaction.
via a series of reactions, we're converting fructose 1,6-phosphate into pyruvate. There's a whole series of reactions there. A whole series of intermediate reactions.
What's the chemical change from glucose 6-phosphate to fructose 6-phosphate? Like, what's the difference between the two?
Don't worry about it. Just know that we've got another sugar, okay, fructose, and it's got 6 carbon atoms.
It's the same formula, right? It's just
Yes.
like a different structure. Okay.
Okay, so in the presence of oxygen, pyruvate will be shuttled into an organelle, which is known as the powerhouse of the cell. So here's an organelle. This is a membrane-bound structure within the cell. And this organelle is known as the mitochondria. Oops. And sometimes you'll see people refer to this as the powerhouse of the cell. So when oxygen is present, so here I'm going to write, above this purple line, I'm going to write W/O2. You're just simply going to interpret that to mean that when oxygen is present, pyruvate, instead of being converted into lactate, is going to be shuttled into the mitochondria. And here, instead of referring to this as anaerobic glycolysis, we're going to refer to this as aerobic glycolysis. And just to be clear, when oxygen is present, pyruvate is shuttled into, just like a PYR there, to indicate that the pyruvate is moving into the mitochondria. Now sit there and think about this. The cell, typical cell, is burning up 10 to 20 million molecules of ATP per second. I'm showing one mitochondria. A lot of students interpret my diagrams very literally, and they'll simply say, oh, there's only one mitochondria in the cell. No. Depends upon the cell you're talking about, but in some cells you may have 25% of the cell volume due to the presence of many, many, many mitochondria. And also, some students sit there and don't realize that here, down in here, we're talking about inside the cell. So this is a very small little membrane-based structure that's known as a mitochondria. here's the cell membrane. If the question is for you to describe the cell membrane, there's a lot to talk about, but one of the most fundamental things you're going to say is that that's a phospholipid bilayer. Here's the mitochondria. And what's not clear, but I'm going to make it clear now, is that there actually are two membranes. So here's a membrane on the outside. Anytime we're talking about membranes, you're always talking about phospholipid bilayers. So this membrane is a phospholipid bilayer. Here's the second membrane, which is a phospholipid bilayer unto itself. So I'm going to come along and we're just going to take a part of this diagram and render it over here. So that little area in the rectangle here is an enlarged view of that. So here's a membrane on the outside, here's a membrane on the inside. They both are phospholipid bilayers. And now I'm going to describe for you the morphology of the mitochondria. I asked this question, I mean, if you went back 20 years or so, you're going to see the same question on every test. So morphology. In anatomy, you were supposed to learn what does that mean, morphology? Anybody, don't be bashful. What's histology? Study of tissue, right? Morphology is a study of structure. Oh, interesting that I have that there because structure does determine function, does it not? So, simple question, what is morphology? You sit there and say morphology is a study of structure.